Welcome everybody to the 5th edition of the knowledge series that is powered by de.NBI, ELIXIR Germany and GHGA. And I will talk about first two networks that support this webinar series. So first the de.NBI ELIXIR Germany network. So de.NBI ELIXIR Germany is a network of 24 partners spread over Germany. You see here the different locations. So I would say we are, completely spreading Germany. And we are, I would say, divided into so-called service centers. So there is a human bioinformatics service center, crop bioinformatics service center, microbial and other. And I would say we are mainly focusing on different bioinformatic topics. So we offer bioinformatic tools and services for the life science. We offer training courses, and we offer a cloud resource that is called the de.NBI Cloud. In addition, we work together with industry, with our industrial branch, and we cooperate also with the European Bioinformatic Network that is called ELIXIR Europe. And the other part is from my colleagues from the German Human Genome-Phenome Archive, short GHGA, and they provide fair and controlled access to data, especially to human data. They are one of the NFDI consortia, and they are mainly coordinated from the DKFZ in Heidelberg. But yeah, they are a federated network of 6 additional data hubs. And they are also here a German node, not in ELIXIR, but in the German initiative, in the European initiatives GDI and the federal EGA. And the basic idea of GHGA is that they want to make genomic research easier and more sustainable. And yeah, on both pages you see the corresponding web pages, and you can also sign up for different newsletters. And now to our speaker of today's session. So Polina. Polina is a PhD researcher from Freiburg, from University of Freiburg. And she's working mainly in the genetics, computer science, and computational biology field. She's interested in AI, and she's a model developer, especially for SARS-CoV-2 proteins. And yeah, she's also a contributor of the famous Galaxy server in Freiburg, and she's developing and contributing bioinformatic workflows and also educational tools there. So Polina, stage is yours.
Good afternoon, everyone. My name is Polina Polunina. I'm a member of Galaxy Europe team in Freiburg, and today I will walk you through DMS data, not only how to experimentally measure variant effects, but also how to computationally extend this data. And the focus will be practical. I will introduce you to the uncertain significance problem and what is DMS experiments and what is DMS data, and how MaveDB stores this data, and that there is a sparsity problem, and the imputation solves this problem. And we feel as a concrete example of this machine learning model that can solve this problem. And we will finish with responsible use of DMS data and future directions. So high throughput sequencing revealed that nearly all single nucleotide variants appear in human population, and millions of genomes sequenced and hundreds of millions of missense variants are identified. But the problem is that most of these variants are variants of uncertain significance, classified as variants of uncertain significance, and for clinical interpretation, functional evidence is required, but it is limited now. Computational predictors like SIFT, Polyphy, and EVE help, but they often disagree and are considered rather as supporting evidence than strong evidence for pathogenic variants. And clinical decision requires stronger evidence. And now we have a situation that we can detect variants faster than we can interpret these variants. So this gap motivates for scalable functional assays. Sequence function approach is one of the solutions. So the traditional methods allows us to measure one variant at a time. So we find the variant in patient, then we test it, and sequence function approach gives us the possibility to systematically test thousands of variants in parallel and gives us quantitative effect maps. And here on this image, we can see the general pipeline of sequence function study. So we start always with variant library preparation, then we deliver this variant library into model system, then we read phenotype, we sequence, and we convert sequencing into functional scores that can be refined to by machine learning methods or other computational methods. Or we can also use this data to train models to predict variant effects. And all this brings us closer to variant effect atlas, which is the comprehensive, standardized variant effect maps over assays and genes. So sequence function studies already helped in cancer research. They support protein science. They help to understand disease mechanism, to prevent disease. They support drug development and precision medicine, and they help with clinical diagnostics. DMS stands for Deep Mutational Scanning. It's a high throughput experimental assay that allows us to systematically test thousands of variants instead of testing one variant at a time. It measures functional effect depending on the assay. It can be fitness, binding, or stability of protein. It produces quantitative variant effect maps, so it does not classify to functional or non-functional variant, but it gives us kind of continuous spectrum of variant effects. An important note is that DMS score reflects performance in a specific assay, and it is not equal to clinical pathogenicity. And in the figure, we can see also the pipeline for DMS study, DMS assay. So we start with a library of genetic variants preparation. We introduce one variant into a cell. So each cell represents one variant. And the next step is the most interesting and most complicated in assays, and there are different methods how we can read phenotype, and here there are a few of assay types shown. For example, selection-based methods like cell growth, when we put cells under selection pressure and we observe how cells survive or grow under this pressure. And after that, we can sequence before and after selection pressure our variants and see the difference in frequencies. Another method is signal-based, such as fluorescent reporters. In this assay, we attach fluorescent tags to variants, and then if the variant degrades protein, for example, then the fluorescent signal in a cell will be decreased, and then we will see the lower fluorescence intensity, and then we sort our cells into beans, and we sequence these variants across these beans, and then we can also conclude how each variant affects this function. And another methods like molecular properties, when we can measure binding or enzymatic activity, or single cell readouts where we measure one cell at a time. But the idea is that we measure the signal and then we convert the signal into frequencies by sequencing. And then these frequencies can be converted into functional scores. And for that, we need to use computational methods. And here I show only one example of computational pipeline to convert frequencies to functional scores. And this is TileSeq-MAVE example from RodLab, which is designed specifically for cell growth assay. And after we compute our functional scores, we can visualize them with the variant effect map, which is a heat map. And in this figure, there is a small piece of gene shown, and each column represents a position in the gene, and each row represents substitution amino acid, and the color scheme corresponds to functional score. So roughly speaking, the red squares represent damaging variants, and blue squares represent functional variants. And there are also cells with no data. So this is like a sparsity problem that we will also talk later about. And this is important to understand what DMS score is. And DMS score is the performance relative to wild type. It's always assay specific. It reflects the phenotype of this particular, that is measured in this particular assay. And DMS score is continuous, so the scale can differ from assay to assay, and also it depends on the computational method which is used to calculate the score. So the scale can differ from dataset to dataset. And often scores are normalized in the way such that synonymous variants are used as a reference for neutral variants, and nonsense variants approximate complete loss of function. DMS is a framework. It's not a specific experimental method. It's a framework, and different assays can be designed within this framework, and I will show a few examples for these assays. So the first example is the protein stability when we want to measure intracellular abundance. So specifically, example of this protein stability assay is when we attached fluorescence tags to the variant, and then we observe how variant affects the protein. If it destabilizes protein, then abundance reduces in the cell, and then we can see the reduced fluorescent signal. And the example of this assay is for BRCA1 ring domain. Another DMS assay example is binding when we want to measure, for example, protein-protein interaction. And the one of the assays is SARS-CoV-2 spike receptor binding domain interaction with human ACE2 protein. And in this assay, we introduce SARS-CoV-2 RBD domain on the surface of yeast. And then we add also a binding fluorescently labeled partner, such as ACE2, and then we see how good is the binding affinity if the variant on this particular cell disrupt the binding affinity, then the labeled partner will not stick to the cell, and then we can also read the signal, fluorescent signal from this cell and make a conclusion how variant affects binding. Another example is enzymatic activity assay, which measures catalytic function directly. And the example here is TIM1 beta-lactamase assay that that is the protein that improves the bacterial antibiotic resistance. And if the variant maintains this enzymatic activity, then we can see that bacteria will grow and survive. And then by measuring frequencies before and after, so this assay is performed under higher antibiotic concentrations, and we can measure frequencies before this pressure and after, and then convert them into functional scores. And the last example that I want to show is cellular fitness, which measures growth under stress. And the example is CHEK2 is complementation assay. So CHEK2 is the DNA damage response kinase that respond to DNA damage. And if we put our cells into the DNA damage conditions, then we can see how variants in CHEK2 help to save this function, or if they disrupt this function. Yes, the framework is the same, and what differs from assay to assay is the phenotype and the biological question that we want to answer. And DMS gives us empirical, high resolution, and quantitative data at scale. However, it has its limitations. So it's assay specific, it's context dependent, it's often incomplete coverage, and there are very few data sets that have a complete coverage. So it's a sparsity problem. And also we have different score scales. So each assay produces scores, and then the scores are normalized in different ways. So we at the end have differently normalized scores, and it's very important to standardize scales among assays, among data sets, so that we can ensure reproducibility. And so MAVE-DB is one effort for standardization of data, and MAVE stands for multiplexed assay of variant effect. This is a broader term for variant effect assays, and DMS is one type of MAVE assay. So in this presentation, I talk about DMS assay specifically, DMS datasets. And MAVE-DB is a community repository for storing DMS datasets. It has standardized format, and it stores these score sets with metadata and enables reuse and reproducibility. And if you are interested in more details, you can read this paper from 2024. And this slide shows a scale of MAVE-DB. So far, there are over 2,600 datasets uploaded into MaveDB, which covers millions of variant scores and over 100 target organisms, and both coding and non-coding assays, even though we can see from this diagram on the bottom that most of score sets are for protein coding assays, but there are also non-coding assays represented in MaveDB. And also we can see that most datasets represented in MaveDB are for human proteins, but there are also different other target organisms represented. So far, I talked about MaveDB, which is the storage for functional scores, but for clinical interpretation, we need additional structure and tools. And here MaveMD comes in. This is a interface that is built on top of MaveDB. It's designed for clinical variant interpretation specifically, and for this, it integrates different clinical resources like ClinVar, ClinGen Allele Registry, and it provides evidence calibration according to ACMG-AMP guidance. It also gives some displays for clinical interpretation, such as assay facts, which is important because context is important for clinical interpretation and clinical performance, where we can see how good data is separated between benign or pathogenic variants, and it provides also a view for evidence strands that is calculated using different statistical frameworks like oddspass or Excalibur. And I will start now to show you the example data set in MaveDB, how it looks like and how to read it, how to interpret the MaveDB record. And this example is for CHEK2 human protein. So to avoid confusion, I would like to explain that CHEK2 name without E is for protein, human protein, and CHEK2 with E is gene that encodes this protein. And red 53 is East ortholog for CHEK2 protein. So this particular CHEK2 East complementation assay measures the function of CHEK2 to rescue growth under MMS induced DNA damage. So MMS is chemical that damages DNA, and CHEK2 is a DNA damage response kinase that can trigger DNA repairment or pause cell cycle. So by using yeast cells with lacking red 53 protein, that is also DNA damage response kinase, we can then induce human protein and measure how it maintains the function for DNA damage response. And yeah, this is the MaveDB dataset, how it looks like. There is a name of dataset, there is a description of assay, and there is also the unique identifier for dataset in MaveDB. And here is how the raw data looks like. This is the table that is submitted by authors of the study to MaveDB, and the required data for submitting to MaveDB is variant and score, and all the rest columns are optional, so they are decided by authors if they want to publish additional information. And Another view is metadata. This particular view is rendered by MAVE-MD, so clinical interpretation interface. So here we can see some data about assay and the context, because context matters. And the next section is abstract and methods. In this section, there is a description of assay that was done in the study. And in method, usually the computational method described that is used to calculate functional scores. So in this particular assay for CHEK2 is complementation assay, authors used this style-seq-MAVE pipeline that is designed for cell growth assay. And the next view is the histogram that shows the distribution of all functional scores. So this view is for all functional scores that are measured in this study. And we can see this shape that most of variants, most of scores are clustered around score 1, which is wild type like or neutral like score, which is very common. And this view can be also used for sanity check if you submit your data set and you can see the very weird distribution, then you can check your computational part or assay conditions. Yeah. And this is the all functional scores that we have in MAVE-DB. And then if I switch to clinical view, then I will have also the distribution of scores from this experiment, but only scores that were already classified in ClinVar by pathogenic or benign. So blue color corresponds to benign or likely benign variants classified in ClinVar, and red color corresponds to pathogenic or likely pathogenic variants. And yeah, we can see that the scale is also differs. So there are much less scores that were classified clinically than scores than the whole score set. And here we can see also that data is kind of separated. So we see the separation between benign and pathogenic, but only seeing the separation is not enough. So we need to map this variance, this scores to ACMG evidence codes such as strong, moderate, supporting pathogenic or benign. And here I show the screenshot from the paper of the study, so I will not go to the mass, but the idea is that authors used the statistical framework such as odds-pass to calculate probabilities of scores being classified as one of these categories. And these classifications are shown in the next view from MAVE-DB. Actually, this is rendered also by MAVE-MD. And here we show the table that shows us how many variants fall into each group of this clinical evidence for pathogenicity. So it can be different groups, normal is for benign supporting or benign strong, and abnormal is for pathogenic categories. And we also see that there are indeterminate variants, and this is also very important and reminds us that we cannot directly classify all variants to pathogenic or benign. And these variants probably fall into the overlapping region after calibration our functional scores with clinical data. And the methods used to calculate these probabilities are described in this paper. So if you are interested in the details, you can easily go to them and read. And the next view that I will show is the intuitive heat map. And here we see the gene, and on the bottom we see positions in the gene, and on the left side we see amino acid substitutions, and squares are colored by functional score, and roughly speaking, the purple square represents a damage invariant, and for example, green squares correspond to functional variants. And if we hover, we can see the detailed information. So yes, and see that there are also many missing variants in this assay. And here is 2D view, but if I click on view protein structure, then I will see the 3D view of protein, because protein is a 3D molecule. And by this view that is powered by AlphaFold predictions, we can see how scores are distributed inside the protein structure. And we can also see that by coloring, that more constrained regions are, for example, inside the core, and more tolerant regions can be in the flexible loops or surface exposed residues. So this is even more intuitive view. And then if you want to use these MAVDB scores, DMS scores in your bioinformatics workflows, you can download them from MAVDB through API or web interface, or there is also a Galaxy tool that is developed particularly for this, to import MAVE data into Galaxy, and then you can integrate it into your downstream analysis. So after downloaded data set, you need to expect metadata, and also inspect how scores were normalized, because as I said, there are different normalization methods used from assay to assay. And if you want to use the scope of DMS data sets, for example, for training machine learning models, then there should be additional normalization performed. And DMS is designed for measuring variants, like all possible variants in protein, but in reality, DMS datasets are sparse, and the reason is low sequencing depth or experimental bias or other reasons. And it's very important to complete our DMS score sets because it's hard to compare across studies if the data is sparse. And for clinical interpretation, it's also important to have more complete maps so we don't miss important clinical variants. And structural analysis would benefit from complete maps. And if we want to train machine learning models, then we need as dense as possible data, because we want to catch some patterns underlying this data. And DMS missing scores are converted to supervised learning problem, which is called imputation problem, and we can train model on observed variant effects, and we can add some features such as sequence embeddings, evolutionary conservation, physicochemical features, and their goal is to predict missing scores and complete our data sets. The earlier approaches were based mostly on evolution, such as sieved, Provine models, and recent models were based more on machine learning methods like random forests, gradient boosting, neural networks, and there are also hybrid models like CPT-1 that combines different methods. And on this slide, I would like to introduce you our VField model, which stands for variant effect field, which we developed and designed particularly for incomplete DMS data sets. It's in some cases outperforming other models, and it integrates different features such as protein language model embeddings from ESM1V, pre-trained model, evolutionary features such as if scores, substitution matrices like Blossom 62, or biochemical properties such as charge, hydrophobicity, and other features. And it outputs predicted variant effect scores. And the motivation to use these particular features like protein language model embeddings and evolution biochemistry is first, we want to capture contextual relationship between residues, and we want to capture selective constraints and structural plausibility, and combining these features together will give us robustness. And we tested our model under sparse data conditions, and we found out that VField models outperform other baseline models under sparse data conditions, such as 20% of data or 30% of data. We already have a pretty high performance of Pearson correlation up to 0.8. But I want to emphasize that these results are based on random downsampling, and it does not guarantee recovery of all clinically important variants. And for this reason, we can determine our future work that systematic evaluation of targeted low coverage DMS designs are needed to use this DMS data for reliable functional scores or clinical interpretation. And if you want to use VField for your data set, here you can find the pipeline how to use VField in our preprint and paper coming soon. And the general idea of the workflow is retrieve data set from MapDB, then preprocess and normalize scores, and then train VField model on this data, then impute missing variants, interpret them, and integrate into downstream studies. I want to emphasize that DMS data should be used responsibly. So computational predictions is not substitute to functional assay results, as well as functional assay results is not substitute to clinical truth. It's always assay specific, and it's depend on context of assay. The assay measures a particular biological function. It does not measure the patient disease causality. So if you deal with patient variants, then you need to follow is ACMG AMP guidelines from Richards from 2015 and recommendations from Brinich and colleagues from 2019. And yeah, remember that DMS is not clinical truth. And as future direction, I can mention cross-assay harmonization. Many assays are and many functional scores are not normalized in the same way. So we have different scales and we want to harmonize assays. And especially for machine learning models and models for imputation of missing DMS scores, we want multi-protein generalization. Many models that exist, they based on single protein, they are trained on single protein, and the generalized model would be beneficial. And we also want to integrate with DMS data with clinical variant databases such as GNOME. And initiatives like MAVDP and MAVDP already started their towards community standards, but we want to improve in this direction as well. And for long-term direction, we want to have a variant effect atlas, which is harmonized and normalized across assays, across genes and contexts. And here is a summary of my talk. So DMS provides scalable functional evidence, and the DMS scores can be saved in standard way with metadata to MAVDP. And the problem in real world is that data sets are sparse, and machine learning models can solve this problem by imputation. And we feel is a concrete example of machine learning model that shows the good performance for sparse data sets. And the important note about responsible application and of DMS data. And thank you for your attention. I have acknowledgements to my colleagues from Galaxy Europe team and also Alan Rubin from University of Melbourne, Australian Biocommons and Galaxy Europe team. My contacts, don't hesitate to contact me. Thank you for your attention.
Yeah, thank you, Polina, for the nice talk, and hopefully see you at one of our next webinars. So also in April there will be the next one. So enjoy, have a nice afternoon, and see you next time.
Bye.
Bye bye.